There we go. All right, so prokaryotic cells and microorganisms. The bulk of what we talk about actually involves bacteria in this course. There is a chapter on viruses, and that, of course, is really germane, really pertinent given our current circumstance. But as far as what we're actually able to do during the regular term, culturing pathogens, culturing organisms, it's usually bacteria. So it's a really very important chapter. All right, so yeah, the first little part of this is I think it probably would have been okay for me to dispense with some of this because you've had it and sort of been beat over the head with it in other courses. But we're talking about living things. We're talking about organisms, right? We're talking about things that are cellular. They're composed of cells, at least one cell. Um, and of course, that was not always recognized. That was something that ultimately, around the early 1800s, early 19th century, a couple of investigators, one named Schleiden and another one named Schwann, took all of the available evidence, all of the available data about the makeup of living things, and arrived at a conclusion, and that was that all living things are composed of cells, right? So they have a cellular structure. And that idea, plus some related things that are part of it, um, is subsumed in this idea that we refer to as the cell theory. Theory is a really grand, well-supported idea in science. It's a glorified hypothesis. But one of the few important theories we have in biology is the cell theory. An addendum to that is that all cells must beget other cells, which ties in with the spontaneous generation idea that was dismantled. It ties in with the idea of biogenesis, that cells have to come from pre-existing cells. Well, that's part of the cell theory. All things are cellular. All cells come from pre-existing cells. And now I think we would add to it that all cells contain some heritable material, that is DNA. They have a genetic code. So all living things have those three things in common. Composed of cells, they came from pre-existing cells, and they all utilize DNA as their code of life, if you will, the recipe that is inherent in all cells that controls their metabolism and other functions. So these cells that we're talking about have within them, they vary in shape, right? But they have within them this stuff called cytoplasm, which is all of the material in there, including a watery sort of suspension of proteins and dissolved substances and various particles floating around. And in more advanced cells, you have some organelles like mitochondria and in photosynthetic forms, chloroplasts. And you have a nucleus in eukaryotic cells, which is the DNA bound by some membrane, actually a couple of membranes. So all of that stuff, all of the contents of the cell, that's the cytoplasm. And always there's going to be some sort of membrane surrounding it. And generally speaking, membranes of all cells are very similar. The archaea sort of depart a little bit from what the other types of organisms do as far as their cell membrane structures are concerned. But for the most part, they all contain phospholipids or phospholipid-like substances and proteins and so on. We'll be talking about that as we go along. So, like I said, there's DNA. There are ribosomes. All cells have to have ribosomes. These are little bipartite pieces of RNA. That is, they're composed of two little chunks of RNA, one small and one big. And those ribosomes are the places where proteins are synthesized. That process, translation, only happens on ribosomes. And so you've got DNA, you better have ribosomes in order to be able to get the information from that DNA into a form that's usable. And then, of course, all cells are going to metabolize. They're going to take things out, take things in. They're going to produce energy, and then they're going to release wastes. So their inputs and their outputs, all of the chemical abilities that a cell has, all of the biochemical abilities that it has, as far as acquiring energy from food molecules or building parts from those same molecules to repair and grow and all of that. Everything is metabolism. Okay? And those metabolic capabilities are controlled by enzymes. Write that in there. So all living things are going to have those. Enzymes are the proteins that serve as the catalysts that drive and ensure that all of these different chemical reactions will happen. So that's important because most living things exist at temperatures that are kind of cool, and chemical reactions don't go really fast under those cool temperatures. But enzymes, if they're present, will allow that to occur, will catalyze those reactions. And it turns out enzymes are proteins. There's a lot of different kinds of proteins, but these enzymes are proteins that control chemical reactions. And proteins are coded by DNA. So there's a connection there. If you have genes, you're going to, through expression of those genes, produce enzymes. And enzymes control everything about how you function. So here's just a little bit of a primer, really quick and dirty, on cell theory and some things related to it. So of the organisms that we have on the planet, they're divisible into two groups based on characteristics of their cells. There are the eukaryotic forms. That's the group that we're part of. And they belong in the domain that actually constitutes a whole taxonomic category, domain eukarya. And then we have prokaryotic cells, which actually consists of a couple of domains. Those would be the bacteria on the one hand, and the archaea on the other. They're both prokaryotic in terms of how their cells are structured. What we're going to focus on in this chapter are the prokaryotes, the prokaryotic cells, and how they're put together. And then in chapter 5, we're going to come back and look at the eukaryote. So yeah, this is just more stuff related to what I was just talking about. All right, so points out that eukaryotic cells are found in these kingdoms. Right, we've got those 4 kingdoms: animals, plants, fungi, and the protists, or protistans. And the thing that sets them apart, well, the two things are, one, they possess membrane-bound organelles like mitochondria and vacuoles and vesicles and an envelope around the nucleus and all of that stuff, Golgi apparatus, ER, all of that. And the nucleus, which is bound by 2 membranes for reasons that we'll disclose when we get to chapter 5 when we talk about the endosymbiosis theory. But yeah, eukaryotic cells have those 2 features in common, membrane-bound organelles and nuclei. Prokaryotic cells that we find in the bacteria and the archaea have no true nucleus. They do have DNA, but it's not bound in a membrane. Nor do they have membrane-bound organelles. They've got some things in there that, like the ribosomes, for example, that some people classify as organelles, but the key is they're not membrane-bound. And then down here, these are characteristics that all living things share in common. You've got cellular organization, and if your cells contain DNA as genetic material, all of those things shown there, heredity and growth and development, responsiveness, the ability to respond to stimuli like changes in temperature, changes in water, availability and that sort of thing. Um, that's responsiveness. What some people call irritability. But yeah, cells can do that. Living things can do that. Then transporting things back and forth across the cell membrane. Metabolism and enzymes is huge. And then reproduction. Cells can produce more of their own kind with great fidelity. And that's because they have genetic material that they're passing on. So it's going to cause the same traits to unfold in their descendants as they have within themselves. So all of those features have to be in place. These are characteristics of living things down here. Those are the what is that? 7 different things that typically are seen in all living things. It's something we all share collectively. And so here is kind of a artist's rendition of what a prokaryotic cell might look like. And this one probably has more parts to it than a typical cell would have. They've tried to pack everything you could possibly imagine into a prokaryotic cell into this one little picture. And there probably are some things that aren't really shown here, but the major things that one would typically need to discuss and know a little bit about if they are if they're trying to show some sort of knowledge of prokaryotes, specifically bacteria. These are the parts of a prokaryotic cell. So rather than belaboring this and going over this image in depth, I'm going to go ahead on because as you see, there's page numbers associated with each of these. This is from an older edition of Tolaro and Chess, so it may not be exactly the same as the version that's out there now, but that's what the book does. It takes each of these parts and kind of elaborates on each of them in turn as they go. So that's a typical bacterium that's sort of rod-shaped. You see how it's longer than it is wide. That's described as a rod-shaped bacterium, or that particular shape is the bacillus shape. Other bacterial cells actually are just little round circles, little spheres. Those are called cocci. Think there's coccus. This is a rod-shaped cell. The technical name for that is a bacillus-shaped cell. Now there is a genus Bacillus. That's an entire group of bacteria that is typified by having that same shape. But we would write that word out if we're talking about the particular organisms that are in the genus Bacillus. We would write that out with a capital B, and we would underline that or italicize it. But the way it's being used here, the word bacillus means long cell. So we have bacilli. That's the plural of that, by the way, bacilli. We have cocci. And then we have, in some instances, cells that have kind of a sort of corkscrew shape. And there's various bacteria like that. And those are called sporili. Corkscrew-shaped bacteria. Those are the 3 major shapes that we have, or what we call morphologies. I'll write that word down here. Morphology means bacterial shape. Anything really. Cells, but all organisms. Morphology. All right. So the first thing, what we'll do is we'll take the cell and we'll go from the outside in. All right. We'll start off with the stuff that's on the outside and work our way on into the inside. Outside, you have external structures. Right. These are things that sort of adorn the cell. They're found as appendages hanging off the cell. And there's 2 things out there. You've got these actual appendages, but you also have on the outside of the cell this surface coating, which is largely composed of carbohydrate, sugars, if you will. And that's what that name literally means, is glycocalyx means sugar coating, sugar sheath. And there's these little packets of sugar-based molecules all over the cell, and those act as markers to identify that cell as belonging to a particular species. They're the kinds of things that our own immune systems would respond to if they were trying to mount an attack to one of these things. They're sometimes referred to as antigens. So the antigenic particles on the surfaces of cells are part of the glycocalyx. And I mention this now first because it's sort of a universal. All cells, including your own, have these. So, but that's something on the outside of the cell. At least a thin layer of those sugars are there, those antigens. In some cases, it can be thickened for various purposes. So we got that out there. Okay, this is non-living material. It's just sort of secreted and deposited out there on the outside of the cell. The appendages are typically extensions that are elongated, and while the glycocalyx is composed of, sugar, carbohydrate, right? The appendages are principally composed of protein. They're proteinaceous. All right, so there's a big difference there. And there's three general classes of these. There's likely some other more specialized ones that we could add to this list, but the main ones are these. So those that are used for motility are going to be used for movement. And bacteria, especially the elongated ones, the bacilli and the spirilla, are actually capable of motility. They can move on their own. And so we'll talk about the structures. There's actually a couple of different kinds of structures that are used for that purpose. Then you have features that are used to give some traction so that the bacteria can attach to a surface and not be swept away by currents of body fluid or water or wherever they happen to be. Organisms can kind of grip surfaces really well using these structures called fimbriae. That's how you pronounce that name. Some people pronounce it fimbriae. If you were to take the E off and write it out as fimbria, you would be talking about just one of these little pieces, one of these little extensions. So, and then we have channels. These are little tubes, again, composed of protein. And those are referred to technically as pili. The singular for that is pilus. So let's talk about what these things are, what they look like, and so on. So flagella are long structures used for motility. They're elongated, in many cases as long as or longer than the cell that they're attached to. And so that's one thing about them. They're typically never really short. They're pretty long. And they are used for movement. They are present so that organisms can literally kind of propel themselves through liquids, right? So wherever these organisms happen to live, there's likely some liquid medium that they're passing through, some water, or body fluids, or mucus, or something such as this. They're able to move through it using flagella. Not all bacteria are equipped with this ability, but probably more of them can do it than you would imagine. Especially, like I said, the rod-shaped bacteria are going to do this. The cocci, as far as I'm aware, don't typically have flagella. Right? They're small, very simple cells, and they lack these. So this is more of a feature of elongated, rod-shaped types of bacteria. And these flagella in prokaryotes are very unique. There are flagella in eukaryotic cells, but they're structurally and compositionally very different. So in a prokaryotic flagellum, you're going to have these three parts. You're going to have for sure the filament, which is really long. And these pictures down here don't illustrate that so well. They've had to kind of cram what they can of the flagellum in there to make it fit. But this thing's going to trail off this way some distance, all right? And the protein out of which that is built is specific to the prokaryotes. You're not going to find this in eukaryotes, but it's flagellum. Right, so that's sort of a marker that would, again, identify the bacteria as being, prokaryotic as opposed to eukaryotic. So that's the same in both of these type. By the way, I did not point this out, but the flagellum shown here on the left is for a gram-negative organism. I apologize for that. It's gram-negative. That's supposed to be a G. Let me write that better. Gram G negative. All right, so that's a gram-negative. This is a gram-positive flagellum, the basal portion of it. Okay, so both of them have filaments composed of flagellum. Now down here at the base, you can see there's this bend. All right, this little hook, as it's called. And that's permanent. That's set that way, and it's not going to bend back out the other way. It's always going to be bent like that. That little curvature in there is really important. Okay, and it's composed of a different protein, and sort of anchors that filament. The filament's stuck down in it, and when this assembly begins to rotate, the hook in that filament will impart a sort of kind of torque on this flagellum in such a way that it will cause the organism to move in either a straight path or in a random zigzag path, depending on the direction of rotation. So we'll talk about that more here in just a little bit. But just know that that hook is essential. If you straighten that out, the capabilities of the bacterium as far as how it's able to move in its environment will be much more severely limited. You've got to have the hook. And then you can see a basal portion. In the case of the gram-negative, you've got a stack of 4 little discs here. All right, they're round and smooth, and they're embedded in the not only the cell wall, but also the cell membrane of the gram-negative. So there's a cell wall that's out here. That's the cell wall. And then there's just a membrane proper here. And in the case of the gram-negative, those little rings, which act as bearings, if you will, are able to rotate inside of the wall in the membrane, and that entire assembly can rotate through 360 degrees, go all the way around at very high levels of rotation, high RPMs, and spin this entire filament. Now, you might think, well, that's no big deal. But think about how many living things you know, regardless of how big they are, how sophisticated they are. How many living things you know have parts that are attached to themselves that can do this, that can rotate entirely? It means that it's not firmly, completely attached. All right? Think about, for example, if you were to try to rotate your arm through 360 degrees, it'd be impossible. You have your rotator cuff muscles and various other connective tissues and so forth anchoring it in place. Likewise, for your head, you can't turn your head through 360 degrees. In fact, no vertebrate animal or any other organism, for that matter, is known to have parts that are capable of doing that. So this has been regarded as a sort of almost miraculous thing by some folks who believe in things like intelligent design. They put forward the notion that nothing like this could possibly have arisen through gradual evolution by natural selection over tens of millions, hundreds of millions, even billions of years. So there's some debate about that. But anyway, that's generally how they're structured. 2 sets of rings embedded in the outer covering of the cell, You've got the hook, and then you've got the filament. The gram-positive organisms have a simpler cell wall, and we'll be talking about that. So whereas in the gram-negatives, there's some of the same stuff. This is called peptidoglycan in here. See that layer of brown little capsule-shaped things in there? Gram-negatives have some of that peptidoglycan, it's PG stuff. But look at the gram-positive. A whole bunch of peptidoglycan and little else, right? And so there's a difference there. It doesn't really relate to the flagellum so much, but I thought I'd take the opportunity to point that out. And you can see the flagellum has its basal body embedded all the way down in the cell membrane. So really not any of it is embedded in the peptidoglycan, not as much of it. So just a couple of rings there. And those too can rotate through 360 degrees. Now here's the thing, with that hook in there, with that basal body, the direction of rotation now is going to make, is going to matter. It's going to have some major bearing on the way that they move. All right, so let's look at that. All right, so this illustration shows a couple of hypothetical examples. This would be a gram-negative, and this is a gram-positive. Gram-negatives being red or pink, and positives being more purple in color. You can also see a couple of differences in these hypothetical organisms here. One of them, the gram-negative here, has just a single flagellum, and so we call that condition monotrichous. You could say monotrichous if you want to accent it differently, but that means a single flagellum, right? And then this guy down here has many flagella. Hey, just substitute the prefix poly in there, and that describes that one, polytrichous. And there's other variations in terms of how flagella are distributed. But in both cases, what you see is the rotation of the flagellum matters. Okay, so in both cases, if as is the case here with the gram-negative, you get this counterclockwise rotation. If the flagellum rotates counterclockwise, the cell will move in a way that is not so erratic. It's really directed and linear, and it tends to go from point A to B more or less directly. And so we call that a run. The same thing is true here in this polytrichous bacterial cell that has many flagella. They're all rotating counterclockwise, and look what they do. They braid together in one big superflagellum, one big mass. And again, the movement is really efficient, and it's directed, and it's straight, and all of that. In these other two examples, the rotational direction has been switched, so it's now clockwise. Okay, so the really nice straight runs are when you have counterclockwise rotation. When you have this sort of erratic, sort of zigzag randomness, that is displayed sometimes, that tumbling, that's when the flagella rotate in a clockwise direction. And look down here in the case of the polytrichous form, each of the flagella are basically on their own. They don't work in unison any longer, but they work separately. And that causes this tumbling to occur. So what if you were to follow these things along for some amount of time? What would you see? You'd see this kind of thing. So this shows what tumbling and running looks like. Here in picture B, they've kind of marked the positions of a bacterium that is tumbling, all right? Kind of just randomly moving around in its environment. So every time it stops, they mark the position, and you follow that for a little while, and you'll see that it's just this, directionless kind of just wandering, all right? Occasionally, though, it'll run and it'll move straight, but for the most part, it's zigzagging around. Now, if you place that same organism in an environment where there's some factor there that varies in its concentration or its, you know, availability from one place to the next, what'll happen sometimes, especially if that organism either favors that substance or is repelled by it, it'll move in more of a directed way, get a lot less tumbling, right? So we call that variation in the amount of something from one area to the next, the sort of gradation in the amount of, say, an amino acid or a salt or water availability, or maybe it's something physical like temperature. The organism moves toward or away from that in a kind of directed way. It's running, and the variation in the substance is called a gradient. Okay? So you can see how it's lightly colored over here, and it becomes darker as we go over here. I mean, some substance is there that is present in the form of a gradient, right? And the organism is moving toward it. So that's kind of what that is. And the substances that they can respond to, as I said, can be chemical. So if you are a bacterium and you're moving toward some chemical substance because you really want that, it's maybe something nutritive and helpful to you, that's called a positive chemotaxis. If you don't like it, if you're running away from it, rotating your flagella in a counterclockwise fashion and going away from that negative stimulus, that's a negative chemotaxis, right? Light can elicit such responses. Some organisms really like light, especially if they're photosynthetic. They'll move toward it. They're displaying a positive phototaxis. Some organisms hate light and can't deal with it. And so they'll try to move to darker places, and they'll run using counterclockwise rotation of flagella to those dark places. That'll be a negative phototaxis. We have these different, and generally speaking, directed movements like that are referred to as taxis, singular as taxis. So chemotaxis, phototaxis, there's thermotaxis, and so on. So that's what running and tumbling is all about. Go back to this. So this shows the different flagellar arrangements depending on the type of organism we're talking about. So here we've got a cell. Notice how this cell is sort of bent. This is a rod-shaped bacterium, but it's bent into the shape of a comma, you know. And organisms like this are described as having a vibrio sort of shape. And there actually is a whole genus of bacteria called the vibrios that look like that. The organism that causes cholera is one of them. And this particular vibrio, you see, has a single flagellum, so it would be monotrichous. And that's typically for monotrichous forms where they come off as at one end of the cell or the other. Not in here like this. They never do that. And then this form is another, it looks like a vibrio, but this time you can see a whole sort of ponytail of flagella hanging off one end of that cell, and that'd be a lophotrichous form. So lopho refers to something in a bunch. And then here's an amphitrichous form. You can see both ends have flagella coming off. There's some amphitrichous bacteria. And then finally, there's this polytrichous form that has many flagella, and instead of them coming off just one place, they come off everywhere. You can see all the way around that cell. This is typical of organisms in the genus Proteus. And then Salmonella is another one. They typically have that kind of, gosh, it's horrible. I'm sorry about that. That's supposed to be an O, and that's Salmonella. Several Salmonella species. So those are flagellar arrangements. And then another type of flagellum is I don't know if it really qualifies as a flagellum or not, but it's used for motility, and it's made of protein. But this occurs only in those organisms that are sort of spiral shaped, corkscrew shaped, and their flagella are internal. Right? So here you've got a chunk of one of these. These are typically really elongated, so they took a little chunk out of it and sliced through that. And if you turn that on in, and if you could have a powerful enough microscope to be able to see what you're looking at there, you're going to have on the outside an outer sheath, and that's composed of I'll just tell you what it's composed of. It's composed of this stuff called LPS, lipopolysaccharide, which is typical of gram-negative. So these spiral-shaped bacteria are some of the gram-negative forms. They are in that group. And then between that and the inner membrane, right, is a space. So here's the inner membrane here. It goes all the way in. There's this space, all right? The cell wall and the cell membrane are not firmly, attached to each other, but there's a space between them. And that space is called a periplasmic space. And you have stuck through that all of these little filaments that in this picture here are cut off. They look like little yellow dots. They're actually elongated, and they spiral and wrap around the bacterial cell, around the membrane containing the cytoplasm. And those proteins can stretch and can contract. So they're contractile. And what they'll do is cause the cell to kind of twist and bend and writhe, sort of squirm, and that's how they move. And so they're not actually contacting the surroundings and pushing against that. The flagella are internal, and they cause the whole organism to twist and writhe. That's what accounts for them. So those are periplasmic flagella. And then finally we have fimbriae, depending on how you want to pronounce it. And unlike the flagella, which are elongated and motile, these fimbriae don't really move much, if at all. They're more static. And unlike flagella, there tend to be many of these, lots of them. All right, so you can see in these pictures here, this is a colorized electron micrograph done with scanning electron microscopy, more than likely. And they've colorized it. You can see this sort of fuzz all around here. Those little short bristles, which occur in tremendous numbers, those are the fimbriae. And they emerge from the cell's surface. And they, along with the glycocalyx material, help the cells to kind of stick to surfaces. That's their main job. And you can see here, this is a picture of someone's intestinal wall. This is actually the top of a single cell. So these intestinal mucosal cells are shaped like this. They're tall columnar cells. And across the top, you got these microvilli. These little tiny extensions of the membrane. And so what you're looking at over in this picture here is a little chunk of the surface of one of those cells where the microvilli are. And you can see the bacterial cells stuck on that surface. And they're hanging on with the help of the fimbriae. That's important for things like E. coli and other gut-dwelling organisms because as material passes through, it tends to cause them to slough off, and they didn't have some means to hold on. Eventually, they would be essentially eliminated. And then the final thing I want to include here in this discussion of appendages are pili, right? And they're made of a different protein altogether. So the flagella are made out of flagellin. The fimbriae are made of a different protein. I'm not sure what that is. But in the case of the pili, there's a protein known as pilin. And like all of these other proteins that are unique or specific to bacteria, Hyaline is specific to bacteria as well. Right? And what it's used for is building a little tube, a hollow tube, which is typically found in the gram-negatives. Gram-negative organisms, E. coli and Salmonella and a variety of other forms that are rod-shaped typically have these. Cocci don't have them, as far as we know. So usually an artist's rendition will have them looking like a little tube that comes off the side of the cell. They sort of stick out perpendicular to the long axis of the cell. And they're hollow. And what they're used for is actually transferring some genetic information from that cell to another cell. So two cells will pair up. Let me change my picture here. Two cells will actually pair up. And they will literally kind of put their pili together. They'll kind of stick them together to make one common sort of conduit or passage between the cells. And then inside here, you're going to have your genetic material. And what occurs is the genetic material, the DNA, which is in the form of a circle, will sort of be replicated. And you'll see a little strand of it emerging as that process gets underway. It's being copied. And what will happen is the DNA from one, we'll call this A, will be passed over to the other guy. B will receive some DNA. And likewise, some of B's DNA is going to go over here to A. And if there's a little bit of a difference genetically between A and B, the result now is going to be some hybridization, this greater variety, diversification of their DNAs through this process. And so that's called conjugation. It kind of looks like sex that we see in higher organisms. The difference, though, is that instead of reproduction happening, those two guys will part ways, but instead of them having the same genetic constitution they had previously, they're going to be altered a little bit. So what this does is it increases genetic variation, which is helpful because this is what allows organisms to adapt to changing environmental conditions. Have to be able to do it. So there are those. And then, the last thing. This is the last thing that I'm going to talk about before putting up another video shortly. The glycocalyx, again, the sugar coating, composed mostly of sugar. There may be some protein in there. But it varies from organism to organism. Now, in most, it's going to be like this. It's going to be a slime layer, which means it's really loosely organized. And you can see it sort of shreds, and it can slough off and, doesn't afford much protection, but it gives the cell some ability to kind of stick to surfaces. So that, together with fimbriae, can be really helpful for organisms. They can stick to surfaces, they can protect themselves to a certain extent. But when they're really challenged, and some organisms do this basically all the time, they'll produce a really thick capsule, which is the same stuff, but it's really consolidated, it's thick, it's dense. And that's going to shield them from injurious kinds of things like antibiotics and organisms that would be trying, or cells rather, trying to kill them, like neutrophils and macrophages. So we have those, and these are common to both rod-shaped, spear-shaped or coccus type of bacteria, and also the spirilla. All of them can produce a glycocalyx. In fact, this is a universal. All living organisms, all cells have at least some form of this. But in bacteria, they're, as is the case in the capsule, much more evident. And so, yeah, this talks about functions of the glycocalyx. So protection for sure is one of those. Protection. So you got a little bit of an extra covering. They're going to experience less water loss and less nutrient loss, but also protection from killer cells like neutrophils and things like that. Cells that are the arbiters of immunity, like the T and the B cells, they can't get at these guys either as well because they have that capsule. And if you got enough of these organisms all sort of glommed onto one another, if they're really piling themselves into a particular area, they'll form a kind of film that can coat an agar surface or the the surface of a drinking fountain, say. You see that sort of scuzzy. It's gross to think about. That's why I hesitate to say it. But if you look around the drain of the drinking fountain, if it's not cleaned properly, it'll have this sort of slimy stuff in there. You can probably imagine a lot of settings where this is the case. That's called a biofilm. And in fact, the tartar or the plaque on your teeth, that's a biofilm as well. You've got bacteria there. And what's allowing all those things to form? Presence of thick capsules. And they allow these organisms to attach. So yeah, over here, this picture A shows an agar surface, and you can see colonies of bacteria on its surface. So these colonies, you can see some of them are nice and tight and well defined. Their borders, don't sprawl and they don't seem to be kind of flowing across the surface. Those probably have a capsule. Other organisms simply have the slime layer, and so you don't see the capsule, but you don't see a nice consolidated colony, but you see one sort of sprawling, as we say, swarming. Different species have different ones. All right, so I'm going to go ahead and stop it right there. Okay, and that's your first lecture on Chapter 4, Prokaryotes. So look for more to come shortly.